One thing that all epidemics have in common is that they strike very quickly. They appear suddenly, like a bolt from the blue. And in this era of medical and technological progress, there does seem to be a tendency to forget just how fragile, how vulnerable humanity can be. At the end of the 20th century, we even believed that we could eradicate infectious diseases entirely.
The COVID-19 pandemic has forced our society into a state of limbo and undermined many of the certainties that we once took for granted. It's a veritable wake-up call, inviting us to reassess our relationship with the living world and take meaningful action to prevent another pandemic from once again bringing our world to a standstill.
The first epidemic outbreaks among humans would probably have been during the Neolithic period, when we first began farming livestock. This new, more settled existence meant that we needed to build an economy that was based on the animal trade and that set certain processes in motion which led to the first epidemics. These may have been smallpox epidemics, measles epidemics. These diseases became endemic.
Viruses are one of many things we share with animals. We can pass viruses on to animals, and though we often forget it, they also pass viruses on to us. Like measles, for example, which developed out of cattle plague. This is all a part of the narrative that we need to be telling ourselves. This pandemic is a global emergency, which means that people are finally paying attention. They're ready to hear this narrative, the story of the Anthropocene era, that is, the series of rapid changes that humanity has inflicted on the environment and the many global disasters that these changes have caused. The idea that a pandemic could happen, that there could be an animal virus to which we may not be immune, this idea really gained ground in the 1980s. This was the decade that Ebola and AIDS were discovered. We realized that infectious diseases were not merely a thing of the past. And in fact, thanks to the accelerating rate of environmental change, especially the boom in industrialized meat farming, it became clear that new infectious diseases were bound to emerge as a part of our new relationship to the animal world. In the 1970s and 80s, when people tried to explain the origin of these new pathogens, they pointed to the revolution in industrial farming, the Livestock Revolution. In other words, the quantity of animals being reared for human consumption was so enormous that it brought 10,000 years of farming tradition to an end. Until then, humans and animals had lived in relative harmony and had exchanged pathogens in a form of parallel evolution and adaptation. But in the 1970s, a threshold was crossed, and we saw change on a similar scale to the changes of the Neolithic Revolution when we first started to domesticate certain animals and plants.
Our impact on the environment means new pathogens are more likely to emerge. But how are these pathogens transmitted?
A pathogen is an organism that lives off another organism and harms it in the process. The first category of transmissible pathogens are what we call vector-borne diseases. That means that the pathogen needs a carrier. It could be an insect, like a mite, a tick, a mosquito, a flea, like the plague, for example. With these pathogens, if you can control and contain the carrier, then you automatically contain the disease. Vector-borne diseases are relatively easy to contain. The second category is what we call direct contact, and the two best-known types of this are HIV and Ebola. The third category is our notorious coronavirus. For example, the flu virus. These are, in my opinion, the hardest to contain because these pathogens are transmitted through aerosols. They can either be airborne, for example, in droplets in the air, or they can pass through physical contact. When I speak, I spray droplets out onto my hands, and if I touch something, I could contaminate it. These categories are a basis to help us understand the ways that pathogens function.
Once SARS-CoV-2 has found a human host, it's able to replicate itself in multiple parts of the human body. There have been a number of scientific breakthroughs that help to explain this process.
We had been working on coronaviruses for the past 15 years, and people started to take an interest in the proteins that we were studying. They also were interested in one of the key components in viral replication, which is the spike protein. It's the protein that covers the surface of the coronavirus. This is what gives the virus its crown-like appearance. We very quickly realized that a change to the spike protein sequence was appearing in a certain location, and this made the spike protein vulnerable to a protease known as furin, which can be found in large quantities in the human respiratory tract. This sequence suggested to us that the virus was likely to be very good at spreading through the human body. What's more, rather than only infecting certain types of cells, it would be capable of infecting many types of tissue, because we know that the furin protease is found everywhere, in every cell of our body.
The change to the spike protein sequence could have been caused naturally or artificially, which raises questions around the origin of the virus.
We went back and reread the studies that were published in the early stages of the coronavirus outbreak. They said that the closest precursor to this virus was a virus identified among a population of bats in Yunnan province, and that the name of this virus was RATG13. When we compared the sequences, we found there was a genetic divergence between them of approximately 4%. Between the virus in the bats, RATG13, the most closely related sequence that we know of, and the virus that emerged in Wuhan province, there was a 4% gap. For those of us who are not molecular scientists, 4% is very, very little. But for the people who carry out these studies on a regular basis, 4% corresponds to around 1,200 mutations. 1,200 mutations is a lot. It means it's been developing for many years. So we started to question whether this virus was truly the closest precursor, as the studies claimed, because we didn't believe it. It was the direct precursor to SARS-CoV-2. In my view, it was more of a distant cousin, and there were missing links between the virus found in the bats and the virus that triggered the epidemic we're now living through.
With the help of these findings, Etienne de Crowley was able to produce 4 hypotheses to explain the origins of SARS-CoV-2.
If this epidemic is a typical zoonotic epidemic, meaning that the bats act as a breeding ground for coronaviruses, and at some point these bats have infected an intermediate host, for example a farm animal, and this farm animal was the carrier that passed the virus onto humans, then we need to identify this intermediate host. Only by understanding the biology of the intermediate host will we be able to take action to prevent further cases of zoonotic transmission. More recently, a second hypothesis has emerged following an accident that took place in Yunnan province in 2012. A group of miners went down into a mine to collect the excrement left by the bats. This excrement is used as a fertilizer, so it's worth a lot of money. All the miners fell sick, and 3 of them died from an unusual form of pneumonia that was very similar to the pneumonia caused by coronavirus. After that, doctors carried out a study hoping to identify the virus and find out where it came from. They collected samples, studied them, and they found that the patient's serums contained antibodies that recognized SARS-CoV-1. As a result of this study, a team of virologists working under Shizheng Li at a laboratory in Wuhan were sent to the mine to collect samples of the virus. They discovered something that, until that point, was considered impossible, or at least unlikely. They found that some of the viruses that were circulating in Yunnan province, that is, the viruses that came from bats, were capable, in vitro at least, of infecting human cells. This was a major discovery because until then, it was thought that bats could only pass the coronavirus onto humans through an intermediate host. Another hypothesis is that the epidemic could have come from intensive agriculture. When you farm animals on an industrial scale, this can lead to certain problems, one of which is that if an epidemic breaks out on a farm, that's the perfect environment for zoonosis to occur. If these farms were contaminated by the same coronaviruses that came from the bats, they could be the source of the epidemic. So one question that still needs to be answered is, could there have been coronavirus infections that went undetected in these farms that were the source of the zoonosis? That's the third hypothesis. Finally, there's a fourth hypothesis to consider, which is the possibility of an accident in a laboratory. As I said earlier, when the teams visited the site to collect samples from the bats, there was a risk that the staff could have been contaminated while collecting the viruses. Once the viruses had been sequenced, one of the aims of this lab in Wuhan was to understand on a molecular level how does a virus that's found in bats suddenly become infectious to the cells of a monkey or the cells of a human. This raises the question, exactly what experiments were carried out on the viruses they had collected? And, in the course of these experiments, could there have been an accident that led to the outbreak of this virus?
Until this pandemic struck, who would have imagined that in the 21st century, we'd still be discussing ideas like quarantine and lockdown? These things feel as if they belong to some bygone age that we read about in history books. But if we truly want to understand it, we need to look more closely at the concept of contagion and the state of emergency that it creates. It's a concept that's closely connected to our understanding of germ theory. In the 19th century, we understood that contact between humans was the ideal breeding ground for a pandemic. But what we didn't understand was how germs were passed from one person to another. Now, thanks to germ theory, we know that these types of contagious diseases cannot develop unless there is a chain of transmission. The only answer is to break that chain of transmission. Another important factor is the environment that our society exists in. Contagion is affected by working conditions, living conditions, poverty, and the toll that it takes on our bodies. To solve the issue of contagion, we have to change our environment. In the late 18th and early 19th century, this was the turning point where we moved the cemeteries out of our town centers because they were believed to be hotspots of miasma and an ideal place for epidemics to spread. We also improved air and water circulation. For example, the roofs were removed from covered bridges to help the air to circulate, and we removed obstacles from the rivers and waterways. All of this produced a relatively coherent corpus of knowledge that actually had a real impact on reducing, or at least changing the way that we deal with infectious diseases. And this was a long time before any treatment had been developed.
It's widely thought that the vaccine is our best hope in the battle against SARS-CoV-2, but researchers are also working to develop medication.
The development of the RNA vaccine is a huge step forward, and it's entirely compatible with the medication. There will always be people who need medication, whether it's because of intolerance, pre-existing conditions, or other risks. At the start of any epidemic, new medications will always be necessary to stop the pandemic from spreading. Whatever their reasons may be, there will always be people who cannot access the vaccine, so it's important that they can be treated with medication.
To develop medication, the lab must examine the crystallographic structure of the virus.
Mapping the crystallographic structure helps us to get a better idea of the mechanisms that allow the virus to function. As soon as you see the visual, it becomes clear. It's like when you open the hood of a car. You see the pistons, the carburetor, and you know you're looking at a combustion engine. It's the same with a virus. You find the virus, isolate its enzymes, examine its structure, and that lets you see how it functions. We found processes in the polymerase, that's the enzyme that synthesizes the genome. We found that some of its properties were totally unique to the coronavirus family, like its speed, because the genome is large, so it's evolved to work very quickly. But even though it's working quickly, it also makes mistakes when it replicates. And that's something that we can make use of when developing antiviral medication. We're trying to find molecules that can insert themselves into this RNA polymerase the same way a grain of salt gets into a machine and stops it from functioning.
Because most RNA viruses use a similar process to replicate themselves, any medication that's effective on one virus can also be effective on others. But until that treatment is perfected, what other options do we have?
In the COVID pandemic, we were forced to use fairly basic techniques, such as avoiding social contact. These were the same methods that have been used for hundreds of years. And as soon as these quarantine measures were implemented, there were people who opposed them, partly because they believed that the measures were not effective, but also because it's a form of coercion. This gave rise to movements that rebelled against the state, and even rebelled against doctors, who were the messengers and middlemen of this epidemic. Even in the 19th century, the debate was the same. Should we protect the economy, or should we protect public health? These debates are all too familiar to us in the present day. There's a long tradition of controversy of this kind.
For a population to willingly accept safety regulations, they must also understand the ways that an epidemic can spread. One way to promote this understanding is with mathematical models that can help us trace, analyze, and predict the epidemic.
How do microbes and epidemics spread? They more or less follow the laws of physics. If you look at how a liquid spreads, an epidemic often spreads in a similar way. This gives us a pretty reliable set of tools to address the situation. Now, there are several components to an epidemic. There's the physical component, the way that it spreads, but you also have the biological component. In other words, how long people can carry the infection, how quickly they recover. There's also the social component, how quickly individuals move around. The coronavirus is an RNA virus, so we sequenced the genomes, and those genomes were shared widely. This is where the principle of parsimony comes into play. So it means that if I infect you, the sequence of the virus that infected me should be very similar to the sequence in yours, because there hasn't been time for any mutations. On the other hand, if I'm infected by someone who's come from India and you're infected by someone from Belgium, then in theory these viruses will be very different. This principle means that we can reconstruct a kind of family tree. Here we have an image of the family tree. Each branch on the tree represents a patient in Europe. The large blue dots denote patients in France. This family tree gives us a clear visualization of the epidemic. One of the tasks our team has been working on is taking this phylogenetic information and using it to extract data about the epidemic. This allowed us to show that the epidemic slowed down in the second half of March. We were able to estimate the length of time that a person is contagious, and you can also see in this graphic here that the virus was introduced to France multiple times. So there's a lot that we can tell from these genetic data, and it's true that there aren't that many teams in France who are analyzing this.
Samuel Elizon is a member of a modeling platform called ModCov-19, which was created at the start of the epidemic, bringing together around 500 scientists from various backgrounds.
ModCov-19 also looked into the situation with the epidemic. I'll give you two examples. Firstly, how to limit interpersonal contact within a school environment. We contacted Bertrand Maury, who is a mathematics professor specializing in the movement of crowds. He looked at how the structure of the timetable affects the school, the locations of the classes, the way the students move around, and the impact that this has on interpersonal contact. Thanks to this model, it was possible to rank each timetable in terms of contact and choose the one that produced the lowest amount of contact. The second example is ventilation in the classroom. How long the windows should be open, and how often, depending on how many people are in there. It was also helpful on an operational level in terms of how to manage calls to emergency services. At first, the ambulances were overrun, so it was a question of how best to handle the calls, how to allocate the hospital beds, especially in the first wave when there were a lot of transfers, and on a more fundamental level, how to manage the personnel in each team. So all of these ways to analyze the epidemic help the authorities to make decisions, to communicate with the public, explaining what was going on, what had to be done to fight this epidemic, and to encourage certain forms of behavior.
With public health, you need to plan ahead, especially when it comes to gathering data. In Vietnam, for example, they've spent 10 years building a database that lets them raise the alarm within 48 hours for certain diseases and provides up-to-date vaccination tracing. Whereas in France, well, a lot of people soon realized what state France's data management was in.
When it comes to infectious diseases, it's the countries of the so-called Global South who know the most. They are all too familiar with them. They don't see them as some kind of rare curiosity. They've learned a lot about them. They're part of the collective memory. They know how to handle them, how to treat them. They don't act as if these diseases just came out of nowhere. Perhaps there's a lesson we can learn from that. Southeast Asia is also a major hotspot for infectious diseases, and they are in the habit of handling situations like this one.
In a world that has already witnessed SARS and bird flu, it's hardly controversial to state that the health of human beings, animals, and our environment are all interconnected. Global organizations such as the WHO are working together to prevent epidemics, but with no contractual obligations holding their member states to account, so far little progress has been made.
The international health regulations, these regulations have a binding power over every state that has signed up to them. That means every member state of the World Health Organization and every member state of the United Nations. This binding power forces them to declare outbreaks of disease, to cooperate in fighting pandemics, and to provide the World Health Organization with access to sufficient information on any emerging epidemics that might develop into pandemics. These are tools that can be used at an international level, which raises the question, were these tools properly used during the pandemic that we're now experiencing? And if so, how? Because it will certainly be necessary to look back over everything that's happened and examine how the regulations were used in the pandemic.
At the individual level, medicine means treating people after they get sick, whereas at the level of the population, medicine is really about preventing people from falling sick. And it was that preventive side to public health, the need to reason with an entire population and not just the individual, that became paramount during this crisis. In a way, it was kind of a wake-up call.
The importance of prevention is one of the key lessons of this crisis, and according to scientists, there are other equally important lessons to be learned.
Right now, people are only talking about coronaviruses, but let's not forget, there are still a lot of other potential viruses that could appear, such as Hendra viruses, Nipah viruses, picornaviruses. Being able to anticipate that is extremely important, and it's not being considered at all. In France, it's not the clinical research, it's the basic anticipatory research that's not being taken seriously. There are geophysical processes in action that are causing the Earth to get hotter. And this presents us with a dual problem, which is that human beings and industrialization are causing an extremely rapid rise in global heating, and animal species don't have time to adapt to this. If it happened slowly, there would be a selection process, and there could be a transition that happened at the same rate as environmental change. But if it happens suddenly, well, a certain amount of damage is inevitable. A lot of damage, really. This is a problem for biodiversity. As for the second system, these days I often ask people, think of an animal. It could be the size of a human or even smaller. Think of an animal that is capable of having 7 or 8 billion individuals on this planet. And 50 years from now, it's likely to be 9 or 10 billion. So if we want to preserve everything and stop destroying ecosystems, how are we supposed to feed 9 to 10 billion individuals without the mass production of protein? It doesn't matter whether this mass-produced protein is animal or plant-based. Either way, they will be biological incubators for pathogens. We need to understand that what we're witnessing is a kind of arms race between pathogens, the way that we feed these pathogens, and the preservation of our environment. It's an extremely complex set of variables, which, in my view, can only be solved at the global scale, with everybody taking responsibility.
We need to re-examine our habits, and I mean all of our habits. Is it responsible to collect guano from mines where the bats are infected with a potentially pathogenic virus? Is it sustainable to keep rearing livestock with the highly intensive methods that are currently used? Does trafficking wild animals pose a significant risk of spreading disease? Is it ethically defensible or responsible to carry out experiments on viruses that could cause a pandemic? It is vital that the scientific community, and even beyond the scientific community, it's vital that all of civilization addresses these questions and reflects on what we do and do not have a right to do with these tools. And that's something we need to accomplish in the near future.